cytochrome P450 is the enzyme that are responsible for metabolism of drugs and toxic material. So today, we're going to talk about the human cytochrome P450 Allele Nomenclature Committee website. By clicking the download protocol, you can see more about the committee. At first, this website is created to encourage scientists worldwide to speak the same language and to avoid homemade allelic designation. This is because it can confuse the nomenclature system and the scientific literature. Plus, rapid online publication and summary of alleles would prevent unnecessary duplication of research effort aimed at characterizing alleles already described. So, in 1999, an initiative was taken to create the Human Cytochrome P450 Allele Nomenclature website. The first webmaster is Michael Oscarson and Sarah Cecil as his successor in February 2004.
The editorial board of this committee include Magnus Engelmann Sandberg and K. Daly and Daniel W. Neberg, whereas the International Advisory Board consists of as highlighted. The nomenclature system chosen for the CYP allele. Currently, the website covers the nomenclature for polymorphic alleles of 22 CYP enzymes which are as highlighted. The CYP allele website is widely used in research and drug development by academic researchers and the drug industry.
So, this is the first database that we want to talk about, which is PharmGKB database. PharmGKB database was established on 2000 to provide information on how human genetic variation impacts drug response. It has a wide variety of information on drugs, including their chemistry, how they interact with different biological pathways to produce a desired medical effect, and how this effect may be changed by specific gene mutations.
Next database is Human Transport Database. We have all the comprehensive gene resources for human transporters. As we all know, transporters are membrane proteins that control the pharmacokinetic process of cellular metabolites. The third database, which is Transporter Classification Database. It incorporates both functional and phylogenetic classification of the membrane transport protein. It also contains more than 10,000 unique protein sequences.
Number 4. UGT Allele Database. The human uridine glucuronyl transferase UGT superfamily comprises enzymes responsible for a major biotransformation phase 2 pathway, the glucuronidation process. The influence of inheritable polymorphisms in human UGT encoding genes has been extensively documented and was shown to be responsible for a fraction of the observed phenotypic variability. This website will discuss novel molecular aspects that may be determinant to UGT phenotypes. Number 5. Pharma-ADME Database. This is the Pharma-ADME database initiated by academic and industrial interests. The aim is to compile lists of genes and genetic variation affecting absorption, distribution, metabolism, and excretion of drugs, and to aid in selection of genotyping assays for product development and clinical trials. There are 3 gene lists, first one being the ADME gene list, which contains genes associated with drug metabolism, the core marker gene list, which is composed of 184 genotypes and 32 genes which have been selected as most important. And lastly, the related gene list, which contains 74 genes that are related to but not involved in drug metabolism, such as drug targets and receptors. Here is a list of the core ADME gene list along with gene symbol, full gene name, and class. And lastly, this is the ADME-related gene list, which comprises of the gene symbol, full gene name, and core marker list.
CYP Allele Database. This is a CYP allele database. It contains CYP nomenclature and CYP allele characteristics. It was organized by the Human Cytochrome P450 Allele Nomenclature website. The main purpose of this website is for management of new allele designation based on recognized nomenclature guidelines, facilitation of rapid publication, and providing a readily available summary of alleles and their associated effect. Next is the Pharmacogenetics of Membrane Transporters Database, or known as PMT database. This database is part of the Pharmacogenomics knowledge base. It focuses on drug response in relation to genetic variations in membrane transporters. So the PMT project focuses on two superfamilies of solute carrier transporters and ATP binding cassette transporters. This database covers coding and non-coding variations as well as both SNPs and indels, but does not provide allele or halotype information.
Helping the kids out of their coats, but wait, the babies haven't been born.